Okay, welcome back, everybody. Hopefully you're feeling good and your brain has rested for a few minutes and you are ready to be wowed again at the wonderful research that's going on here. But now we've got a new round of contestants that are also really excited. I asked, maybe it was the first round, I said, how are you guys feeling? Are you excited? And they're like, well, that's not the word I would use. But I do want to take a moment and make sure we're still engaged. And so I'm going to ask everybody to stand up again. I know, yeah, don't get comfortable yet. The judges are getting their pens and papers out. You're gonna need your hands for this one, yep. And you guys, you know the drill, you're up and ready. Okay. So I would like you to, if you got anything in your glass, hurry up and drink it fast or find a safe place to put it. And what I want you to do is I want you to find a partner, but not someone next to you. I want you to look around the room and, you know, it's like some enchanted evening and catch someone's eye. Okay. If no one's looking your way, then jump up and down somehow. You don't have to get close to them. You can stay where you are. Okay. And with a lot of science communication and communication in general, one of the things that is key to being understood and to understand is to have that back and forth between the speaker and the listener, right? And so when people who do this sort of thing professionally and get up in front of people, they learn ways to engage with their audience. Lots of times we as science communicators do not learn those ways, and so we are left to our own devices. But one of the ways to engage with your audience is to make sure that you are seeing them and that you're taking their feedback. And if you need to adjust your message because it's not landing, then you do that. And when I teach my undergraduates at 3 p.m., lots of times I'm making adjustments. But right now we're going to do an activity that practices that. And so I want you to get your partner, and one person is going to be the person who's throwing something. So decide who that is. Okay. Okay, so you've decided, and you're going to be the first person who throws it. The other person will catch it, and what I want you to do is I want you to act as realistically as you can like you're tossing a ping pong ball to your partner. Okay, and your partner needs to catch it as if you would catch a ping pong ball. Okay, go ahead and do that a few times. So the size, it needs to be size appropriate. Okay, very good. All right, some of you look more athletic than others. Next up, I want you to act like you're throwing a bowling ball. Okay, so heft the bowling ball. This is like a 12-pounder. And throw it, and I want you to throw it in the air and catch it. That's right. Oh, gosh. Okay, very good. And lastly, I want you to do a beach ball to each other, okay? So go ahead and toss the beach ball. All right, very good. Go ahead and give yourselves a hand. You guys are wonderful ball tossers and catchers. This is also a great activity if you have boys that are, I have 2 young boys, and they always want to throw or kick something in the house. And so, yeah, right. So that is, I like to do that activity because it teaches us and demonstrates how much what and where the message lands, like depends on what we're doing as the presenter, in addition to what you're doing as the audience. And so there are lots of friendly faces in the audience today, and I'm grateful for you guys for being here and supporting these folks that you will do now in the second half. So we'll go ahead and get started. From the School of Marine Science and Policy is Sarah Sweat. Sarah? She's not here. So that was a good practice. Next, from the Department of Entomology and Wildlife Ecology is Meredith Lewis. Meredith, the time is yours.
Owners of a building in Chicago found themselves in a bit of hot water on the morning of October 5th, 2023. Lights had been left on during a large migratory bird movement the previous night. That morning, nearly 1,000 birds were recovered after they collided with the building as they ventured south. Now, this number is staggering on its own, but for those of us who want to know more about the lives of these birds deeply, there was not just the question of how many collided, but of who collided. I scanned photographs that morning and noted some familiar faces: palm warblers, magnolia warblers, soras, and noticed the curious absence of others. Why is this the case? Is it route fidelity? Are some species more vulnerable than others? Or is it simply a matter of wrong place, wrong time? In bird migration research, we often estimate the numbers of birds flying high in the air without necessarily knowing a lot about their identities. My research uses automated recording units and new AI detection algorithms to listen to the community of birds flying high in the air to start to get at that question of who is up there and how do they respond to light pollution. I am also looking at how factors such as habitat and light levels affect how these birds aggregate together or when they stop over on the ground. With this information, perhaps we can more effectively plan efforts to turn off our lights, identify vulnerable species, and make those highways in the sky a little bit safer. Thank you.
All right, thank you very much, Meredith. You guys, your jazz hands are so good. Next up, from the Department of Chemical and Biomolecular Engineering is Austin Morgan. Austin, the time is yours.
In the 1993 film Jurassic Park, Jeff Goldblum's character had an iconic line: "Your scientists were so preoccupied with whether they could, they never stopped to think whether they should." Today, genetic engineers are making modified bacteria capable of fighting our pollution crisis by degrading plastic, and engineered fungi that can clean up freshwater resources by removing toxic contaminants. For these solutions to be most effective, they require the environmental release of engineered microbes. It's something that we could do, but is it something that we should do? Many scientists think it is, but acknowledge that we need safeguards against unintended consequences. A released microbe could be pathogenic for an animal important to its ecosystem, resulting in severe unintended ecological disturbances. Ideally, we would build off switches into the organisms that we release, allowing us to deactivate them once they've accomplished their purpose. But for an organism spreading in the environment, how do we find it to activate this off switch? Instead of looking for a needle in a haystack, we'd be looking for a needle in the Amazon rainforest. My research aims to solve this problem by developing a new tool for biocontainment called Time Bomb. Time Bomb is a programmable biological timer made of DNA and enzymes. We could put this timer inside of a genetically modified organism where it would silently tick, allowing the organism to spread and help us clean up our environment before ultimately triggering an off switch in the form of a toxin that's produced to kill the organism. If my research is successful, it could enable the widespread yet safe deployment of engineered microbes capable of helping us solve our most difficult environmental challenges. Thank you.
All right, thank you very much, Austin. Next up, from the Department of Biological Science is Krishna Thakkar. Krishna.
Okay. Good evening, everyone. As we entered the reception today, all of us enjoyed the warmth by the fire. We feel warmth of the fire from far away, but as it is close to us, we feel the burn. So today I'm going to talk about a thing which puts out fire: water. We have been using water bodies as dumping grounds for a long time. It's not a new thing, and we have been dumping plastic. When this plastic breaks down into small filaments, which are less than 5 millimeter, it is called as microplastic, which is not visible to our eye. But this microplastic gets lodged into the muscle of the very fishes that we eat. This plastic has been seen to cause neurotoxicity in such fishes. Now imagine the bioaccumulation and biomagnification happening in the food chain because of that. But this problem of water is actually on fire because it is closer to our home. In the hiking places like Boogie Run and Curtis Mill, our study, which happens every year with collection of water samples in the spring semester, sees how this microplastic content in the nearby water bodies has been increasing, which decreases the macroinvertebrates, chlorophyll A content, and algal species. So I can say that the water issue is actually on fire, and today when you go out, remember when you take the sip, are you drinking water or plastic? Thank you.
Thank you, Krishna. Next up, from the Department of Plant and Soil Sciences, is Kendall McCoach. Kendall, time is yours.
Forests store carbon, and this forest carbon storage is a widely adopted, government-endorsed, nature-based solution to climate change. But as cities expand, more forests are subjected to urban conditions. So how does urbanization impact forest carbon storage capacity? Fortunately, in this room today, we are surrounded by forests which have been embedded in our urban ecosystems for decades, sometimes even centuries. And the trees in these forests record history. Every year, trees produce a ring of new growth around their trunk. The width of this ring represents the tree's response to growing conditions like climate. Studying the variation in these rings throughout time tells the story of how this tree has responded to changes in climate and urbanization. By combining this analysis with tree inventories, I can produce detailed estimates of forest biomass, a proxy for carbon storage. This provides a much-needed understanding of our urban forests, which is so important because they comprise the majority of forested area across developed landscapes like the East Coast. Ultimately, we have decided to rely on forest carbon storage as a solution for climate change. So we need to understand the capacity of the forests that surround us to provide that service. Thank you.
All right, thank you, Kendall. Next, from the Department of Geography and Spatial Sciences is Manan Sarupriya. Manan, the time is yours.
Thank you. The east coast of the United States is experiencing sea level rise at a rate twice the global average, putting our coastal farmlands at a risk of a silent threat of saltwater intrusion. Imagine unwanted saltwater creeping into the very farmlands that feed us. It's like a domino effect that hits your plate directly, where climate change is also making it worse by introducing more severe and frequent storms, floods, droughts, which makes it all the more important to have a very sustainable management and carefully monitoring system for these farmlands. My research uses satellite data and machine learning to build a model which acts like a smartwatch for these farmlands. It monitors where is saltwater intrusion occurring, which is equivalent to a human health issue, and it also measures the progression of it in both space and time. It's like a constant check on the pulse of your food sources. Previously, it was very difficult to monitor saltwater intrusion because it's very tedious and it requires for the person to be on the field most of the time. We are solving this with our groundbreaking method that uses freely available satellite data that covers the entire Earth every 5 days, giving us high-resolution information without putting our boots on the ground. Our method is computationally very inexpensive that makes it usable for researchers from developing nations as well. We are securing what lands on your plate 1 pixel at a time. Thank you.
Thank you, Manan. Next, from the School of Marine Science is Tianyin Ouyang. Tianyin, time is yours.
Submarine groundwater, the water from continental aquifers into coastal ocean, impacts the surface water quality and geochemicals by transporting nutrients, organic carbon, and contaminants. It's also a large but understudied contributor to climate change and can release up to 13 teragrams of highly labile organic carbon to the surface waters annually. This exported labile organic carbon tends to be rapidly converted to CO2 in surface, contributing to 1.77 million metric tons ton CO2 in the US, which is greater than 1/3 of the 23 major sources reported by US EPA. Locally, in Delaware inland basin, submarine groundwater accounts for around 80% of freshwater inputs and hypothesically to transport abundant nutrients and organic matters to the surface water bodies, which likely causes eutrophications, hypoxia, and toxicities in surface water. My research, therefore, studies the geochemical cycles of submarine groundwater in the inland basin and will thoroughly answer how it impacts climate, water quality, and our coastal life in general. The outcome of my research will be compared with international data sets to develop a conceptual model that benefits the water source management and Clean Water Acts in the US and more broadly. Thank you.
Thank you, Tianyan. Next, from the Department of Entomology and Wildlife Ecology is Destiny Mann. Destiny, the time is yours.
Thank you. Huge problems face the world population today, such as poor waste management, expanding insect species that are invasive, and the struggle to provide a sustainable protein for the rapidly growing population. As an example, in 2018, 63 million tons of food waste were thrown out from US households alone. Of that 63 million, less than a third was managed in any way, recycling or composting, and that left 40 million tons going directly into our landfills. With such huge problems, I think that if we want to manage and understand these problems better, we need to think a lot smaller. Edible insects. So in Europe, 4 species have already been approved, and that's the migratory locust, the house cricket, the yellow mealworm, and the lesser mealworm. The lesser mealworm is actually approved recently in this year and is a common poultry industry pest. If we're able to use these poultry and pest life history traits, like being able to reproduce them in very cramped spaces, in low nutrient available environments, and with a very quick life history, then we can actually reduce waste and produce a sustainable protein that can be harvested at the same time. So my research is optimizing this system. So I'm going to feed them a variety of different feed waste substrates, and I'm going to determine which waste is actually going to be reduced the most while producing the most protein during growth and development.
All right. Thank you, Destiny.
Thank you.
Next, from the Department of Materials Science and Engineering is Shai Santiparat. Shai, the time is yours.
Have you ever wondered what would it be like if you could use solar energy everywhere, not just from the shiny blue rooftop on your house? Let me introduce the flexibility to the world of the clean energy. There's a material called perovskite, low cost, less polluted, easy to make, and the most important thing, it can be made very thin. Looking at your hair, it's pretty thin, right? Perovskite can be made 100 times thinner than that. And that allows flexibility that can be mounted on any curved surface, such as on the car, or even on the fabric. That's impossible for the conventional solar cells. Now, if perovskite is so great, what are we waiting for? The problem is with the scalability. Right now, the conventional method that we use to fabricate perovskite is for the liquid-based method. And that allows only a small size of solar cells. And if we increase the size, it's going to lose the uniformity very fast. And it's where my project comes to play. Now, I'm using the vapor process called vapor transport. So instead of using a liquid-based material, I'm using a gas-based material that helps spreading the layer more evenly. And no matter how large the material I make, it's not going to lose uniformity. And eventually, this method is not going to just stuck here in my lab. It's going to be a game changer to the world of renewable industry. Thank you.
All right, thank you, Shai. Next, from the Department of Plant and Soil Sciences is Jack Dietrich. Jack, the time is yours.
One of the biggest threats to biodiversity here on the East Coast enters our ecosystems in the smallest and most inconspicuous way. The seeds of many non-native plants disperse into native ecosystems, most often through plantings in our gardens and parks. Because cities have the highest concentrations of human activity, native ecosystems within these cities, such as urban forest, are the ground zero for many of these invasives to establish and spread. So if we can predict which invasive plants may spread their seeds into these urban forests, we can not only mitigate biodiversity loss, but also prevent the further spread of these non-native plants. To do this, we can look to see what seeds we find within the soils of urban forest, giving us a sneak peek into the potential future of these ecosystems. We did this by sampling soils from 25 urban forests between North Carolina and Massachusetts. We brought these soils to greenhouses where they were monitored for germination, and we identified and counted each seedling that emerged. About 1/3 of all of the seedlings that we saw were from non-native plants. And in some of our forest plots where we saw one invasive tree, over half of all of the seeds from these plots were from that single invasive species. So we hope our study can help us understand how invasives become established in forests, and in turn, how we can maintain biodiversity within our cities. Thank you.
All right, thank you, Jack. Next, from the Department of Chemical and Biomolecular Engineering is James Buchan. James, the time is yours.
If we stop fossil fuel production today, we're going to continue emitting CO2 from vital industries such as agriculture and construction to the tune of 1 billion tons per year. We will need to be able to offset these emissions to meet our climate goals. And one of the technologies considered is direct air capture. Unfortunately, this is an expensive technology at $1,000 per ton. If we think about that at scale, we're talking about a $1 trillion per year price tag, something larger than the US military budget. The US has recognized the needs for this and has started providing research funds for research such as my own to reduce that cost to $100 per ton. When we started research, we looked at the current state of affairs and saw that the energy costs of current technologies exceed that budget, and we knew that we needed to try something different. We found nickel hydroxide from AA batteries could be a solution, a technology that's been around for decades. With this in our electrochemical cells, we can produce hydroxide which reacts with CO2 in the atmosphere to make carbonate. This can be separated and concentrated and sequestered underground. In our labs today, we have been able to get our energy costs down to $50 per ton, meeting our goals and something that we're looking to improve and scale to meet the direct air capture needs. With this and many of the innovations discussed today, perhaps we'll meet our climate goals.
All right, thank you, James. Next, from the Department of Civil and Environmental Engineering is Harrison Fleetwood. Harrison, the time is yours.
California taxes, New Jersey drivers, vegans. What do they have in common? A less than perfect and somewhat deserved reputation. Fracking suffers from a similar issue of public perception. What if we could bring a win to one of oil and gas production's least popular practices? One byproduct of fracking is produced water, a very salty, briny liquid created as a fracturing fluid. When it accidentally spills, it kills plants and microbes and degrades soil quality. The current remediation strategy: excavate, landfill, and replace the contaminated soil, coming at a great cost to both the environment and the reputation and bottom line of the oil and natural gas producer, a lose-lose situation. My research explores a new remediation strategy that can flip this issue into a win-win. When produced water spills, water evaporates up through the soil, while salts precipitate under the subsurface. These trapped salts are what create the dead zone in the contaminated soil. My research seeks to use crystallization modifiers to disrupt and delay the precipitation of salts. More simply, this means we aim for the salt to form on the surface of the soil rather than beneath it where it can be cleaned off. This in situ remediation technique preserves the original soil while forgoing the heavy machinery required to excavate and transport contaminated soils. Oil and water don't mix, but oil and natural gas production and cost-effective, environmentally sustainable remediation practices can. Thank you.
All right, thank you so much, Harrison. And thank you to all of the presenters from this half. That wraps up this half and the evening. Let's give everyone a big, loud round of applause. Fantastic talks. For everyone here, if you go ahead and scan this QR code, you get a vote too. So this will be the fan favorite. You scan that QR code and you'll have the opportunity to vote for your favorite. So we will now, the judges will finish wrapping things up, and then the DENIN fellows will tally up the scores, and while they're hard at work, trying to avoid any hanging chads, we will, most people don't get that, the kids don't get that, right? We will move out, and I hear that there actually are hors d'oeuvres now, yes? All right. So let's go ahead and give these guys and gals another round of applause. And go ahead, folks, and you can make your way out and have the hors d'oeuvres. Thank you again.